Exact position of the QTL we can't determine because we are
Yeah,
not taking marker, means two marker, flanking marker, we are not
yeah,
taking only one marker.
e- exactly. Exact position of the Qtl marker you don't know. See, for example, this is single marker analysis. This actually, what you are looking is single marker analysis. The model we will going to develop is like this. For example, your the phenotype is equal to marker genotype plus environment into sorry QTL, the probability of getting QTL, marker phenotype and the genotype and the environment. Like this, there will be one regression model we develop. One model we have to develop so that every covariate should fit into this model. Like for example, x is equal to f of y. Like this, there will be a model in I think in mathematics we will study like y is equal to f of x. If you put x, if you put x is a 1, there should be y is equal to 2. There will be some models we develop in our mathematics. We will develop say model like y is equal to f of x. For example, a is equal to x plus a, x plus y. Like this, there will be simple algebraic model how you will develop. Na? In the same way, it is in a computer also. You have to feed in a model wise. For example, you take is a y is a phenotype, if x is a genotype. Okay, if you provide a genotype here, there should be some phenotype. Like this, there will first you need to develop a model. Then, according to this model, the software will calculate everything. Then it will give some product. This is the only simple logic. No need to remember all the thing. But everything is not only Qtl mapping. Almost all the thing which is revolves around regression. Who given the term regression? Like who first time used the regression?
Pearson.
Not Pearson. Carl is not called Pearson.
Fisher,
Fisher,
I
sir.
think.
Fisher, I think. Search it because I am also confused. Fisher, I think. Um, and Francis Galton, I think many of it you know. I think Galton given regression.
Yes, sir. Galton only.
Yes, Galton.
Yeah, that's why Galton was. Then he's a famous, but I think geneticist also famous. He how he developed the regression models. So this is everything is revolves around regression. Then, for example, as you told, we can't determine a position in the single marker analysis. And what we will do in single marker analysis, for example, you just forget this marker now as of now, and this you forget. This marker 1 and QTL will be there. And there will be, and the marker is present at particular distance from the QTL. For example, you think if the recombination will be the more and more and more, what you're going to get, there will be this effect on your relationship between the marker and the QTL. Means, for example, you identified this marker to this QTL. Okay, this is significantly associated with the QTL. But at what percent of recombination, it's very difficult to understand because we don't have linkage map here. We don't have any linkage map. We don't know at what distance the particular marker will be present from the QTL. And if there is any more distance, definitely it affects your QTL results. That is confounded with the effect of the recombination. More will be the recombination. Like normally should be very near. After getting, we'll get nearer markers. But the distance will be more, then it will affect your results. That is the one point. Then, but if you know the linkage, because we here you don't know the distance, how much distance, and at which side also. Either this side is there or marker this side is there, we don't know because we don't have any linkage map and we don't have any distance between okay marker 1 marker 2 how much distance is there, we don't know anything. That's why this is basic method is not used for any for like precision QTL mapping. This is about a single marker analysis. Then simple interval mapping. What yesterday we discussed, but overall in nutshell I have discussed, but we will look at very in detail. For example, you can check marker 1 and marker 2. Here, how you calculate? Here, see, the lot score is very, very less. Marker 2 and marker, see, this one. The lot score is quite here. The marker 20 and marker, this interval you take the lot score here like this. Marker 5 and marker 9, lot score is like this. See, this marker, between marker 13 and marker 8, there will be some peak. It is crossing this threshold. And here marker 8 and marker 8 is also crossing threshold. See, up to here. And up to here it is crossing the threshold. Means from this point to this point, there will be a QTL. At a single point of time, we are just assessing whether QTL is there or not. Like this, we are going. Actually, it is only. And this is confidence interval. From which position to which position your QTL is present. That is called as confidence interval. We'll discuss in detail what is confidence interval definition. This is what single marker analysis. First, you have to check the model will check between marker 1 and marker 2 either QTL is there or not. Then this marker and this marker QTL is there or not. And this marker and this marker QTL is there or not. This marker and this marker means shifting. It shifts the window, but one window at a time. But in other inclusive composite interval mapping, it will look all the window at a single time. The software, like the capability of the software is increases because the time will develop, the computers are developing, supercomputers are developing. Based on that, your software is also like you take the mobiles. If you buy one mobile at this time, there will be a different processor. In next year, there will be advanced processor, which is being easy to use, easy to have some. See now with the face only, it will open the lock. If you just flip off the mobile, it will the lock. It will become silent. How it is developing in the same way. First, they have software only ability to look at one window at a time. But now they have inclusive composite interval mapping. They can look at multiple interval at a time and they can check how they are interacting. That's why they can able to identify the epistatic effect. If you remember this much differences and what is log value, that is more than enough. Then how we can see that marker 1 and marker 2, if you create the linkage map, we know the distance r. But from marker 1 to this QTL R1 plus from marker 2 to this QTL R2. Actually, this is a lot. This is how it will going to calculate. And accuracy. Accuracy is very, very important here actually. Because when you have a single marker, the error will be more. But for example, if error is like for marker identification of the error will be into like 0.1 error will be there. If you're having 2 in this one, it will become 0.1 into 0.1. M into 0.1 into 0.001. Means here there will be 1% error, but it will become 0.1% error in terms of percentage. Means if we have a 2 marker, saying means your confidence of saying this is a true will be increases. But when you're having only 1 marker and you are saying, okay, my QTL is there, error will be more. But if you're having a 2 marker and deciding the QTL, then your error will be very, very less. This is one of the advantage of simple interval mapping. Means you will get more precise QTL. We'll get more precise QTL. More precise QTL in simple interval mapping, but affected with ghost effect. For example, here on QTL is there, but here also some QTLs were there. Because of this QTL, maybe if you use, this is actually because of composite interval mapping, you are getting single 3 QTL peaks. 1, 2, 3. But if you are using simple mapping, instead of getting 3 QTL, you may getting single large QTL. Here the PV will be like 5, 10, 15 like this. But if you use simple interval mapping, even though there will be three different QTL, but you will get a single QTL with more effect. Okay, this QTL maybe you are thinking, oh, I got a large QTL. It will revolutionize your breeding. You'll think like that, but it is not like that. But maybe this will be a goal because of this overestimation because of the ghost effect of the neighboring QTLs. But if you use composite interval mapping, it will give a distinct QTLs, distinct peak, three separate peak. But if you're using simple interval mapping, single peak. That is the only difference between composite interval mapping and simple interval. How it will do? Yesterday we discussed. Okay, understood? What is SMA? What is simple interval mapping, composite interval mapping? Anybody have
Yes,
a
sir.
doubt? Yeah. Then normally in human hand we can't do this. We use a softwares. Regularly use this ICIM mapping, ICIM QTL mapping. Even I also use the same thing. We can use SNP data or SSR data, any marker data it can able to fit. First it will do ANOVA, then it will it will create the maps, it will create the QTLs. Okay, it will do all the things. Okay. You just keep on uploading. There will be one manual also here. I think there will be help. You open the help, there will be one manual. It will give everything. How to arrange your Excel sheet, how to arrange phenotypic data, how to arrange your genotypic data, how to give information of the marker, chromosomes, linkage groups, then how to arrange one after the other, and how to add. If you are using real population, what code you have to use. If you are using NAM population, what code you are use. If you are using CSL, chromosome substitution lines, what QTL you use. If you are using multi-environment data, what code you have to use. Everything. And if there any segregation distortion will be there, you have to clip. And you can map consensus map construction. For example, we have QTL studies from different paper 1, paper 2, paper 3, paper 2. Like you can create all the linkage maps in multiple QTL studies to a single study, creating a consensus map. Like QTL meta-analysis it is called as. There are n number of things we can do in this software. And how Excel sheet will be look like, I will sharing another file.
Sir, it will do simple interval mapping or composite interval mapping.
All things it will symbol all the things simple interval mapping, composite interval mapping, SMA. It depends. What option you will give, it will give all the things. Wait. For example, these are the markers. It's visible my screen.
Yes, sir.
Yeah. These are the markers I have used in the rice. Let's see how many markers. See, like 156 polymorphic markers. Then actually this is a QTL survey study, but I will show. Also, then how to create maps. Like this is how to create the map. This is not required. Okay. Then, for example, these are the marker data and how your genotypic data will look. We need genotypic data. For example, this is chromosome. Chromosome 1, chromosome 1, chromosome 2, chromosome 2. Like this, you have to arrange the chromosomes. Then you have to arrange your primers, like markers, like this marker 1, marker 2, marker 3, marker 4, marker 5 in different columns.
Sir, this is
Then,
SSR marker.
yeah, this is SSR markers. Then these are the individuals. These are the mapping population. 1, 2, 3, 4, 5, 6. These are mapping population. Based on the gel I have shown a picture how to score. For example, in first individual, it's parent 1. This parent 1, parent 1, parent 1, heterozygous, parent 2, heterozygous, parent 2, heterozygous, parent 2, parent 1. Minus 1 means the band is not there. Like this, you have to score the individuals. Okay. Then after scoring, I have to do if you genotypic data. Actually, I don't have phenotypic data here. There will be another file phenotypic. Actually, it is not the actual file. We need to feed software. It is a genotypic data. You take select this sheet, and there will be same. Here, how the code will be there. Na? For each trait, there will be a phenotypic data. Like I think this is single marker analysis I did in ICIM mapping. For example, it will give results like this. Trait, it will give for trait which, for example, these are the traits I have used. Okay, average gall and average egg mass and average egg mass per egg, average multiplication factor. These are the different traits and on which chromosome and which position, which marker and LOD values. See, for each marker it will give LOD values. Because I am using single marker analysis, for each marker it will giving LOD values. Then phenotypic variance explained also it is giving. Then what is additive effect, what is dominance effect, everything it will give. Just you have to upload your genotypic data, like you have to score and you have to create the genotypic data like this. Then there will be same way another Excel sheet for phenotypic data. If you upload in ICIM software, it will give a nice data like this. Then whatever things you can do, it will, if it is any QTL simple interval mapping, it will give like peaks and everything. Okay, like this type of peaks. This type of peaks they will give. Okay, then everything the Excel sheets also give on which marker to which marker it having peak and what is the phenotypic variance of this QTL. Everything it will give and what is the log value for this peak. Okay. No need to do extra efforts. Like you can make the tables, how will it looks like I have presented it in table. Like for each trait, there will be log value, PV value, log value, PV value. For example, normally PV value at least more than 10%, there will be a major QTL. Here, actually, this is the QTL survey. I didn't find any QTLs from these populations. Okay. This is how we have to do a QTL mapping. Then, what are the different softwares were there for the
Sir,
QTL mapping? Hello?
sir, you didn't found any QTLs
No,
in
no,
the
no. Actually,
population?
that is different survey. Actually, that is QTL survey, QTL validation survey. I have published a paper. I will share that paper. If you are interested, you can read it. Okay.
Okay, sir.
That is QTL validation. Actually, I took reported QTLs from different papers, and I checked
Meta-analysis,
either
sir.
that QTL is present in the my uh, that QTL is present in the my population or not. I have checked it. Okay,
It's
but this is
meta-analysis,
a
sir.
very good important study. Hello?
It's meta-analysis, sir. QTL
Yeah, it's
meta-analysis.
kind of a meta-analysis. It's
Sir, but
meta-analysis.
they will also spend more time on the genotyping now, so then it will be some.
No, actually, we are going for QTL mapping. That genotypic data I showed you, not only that is used for QTL mapping also. Maybe there will be a new QTLs. Actually, we have that is only few markers. We have screened 2000 markers. My SRF actually along with me one SRF is working under my guide. He actually doing for his PhD. Like he used to screen 2000 markers. He got nearly 250 markers. He got QTL at fifth chromosome. Like still there are many things he did actually. But as of my study, it is just a validation. It's my second objective actually in my MSc. That's what I did. That's mine is actually different work. That's why I didn't focus much. But I got very good paper from that. Yeah, it's nearly NAS rating about 10 actually. See, and one thing I had to tell you, like many people will say my work is not good like this. Like many people you are telling like work is not good, how either I can get paper or not. The paper writing is always art of writing actually. How you'll present your data, It is more important actually. Either your work is simple or large, but how you present that, like how you interpret it. More the interpretation will go more for good journals. Okay, if you don't have only if you have molecular work, then only it published, or only field work there will be published. It's not like that. First, should aim for the journals which is very very specific for your objective. If you have only field work, opt for the general like plant breeding, field crop research. Okay. If you have molecular work, there will be a separate journals for molecular work, molecular biology reports. Like there will be aims and scope and the way you interpret. That's more important actually, rather than how much you do. Okay. Then there are different softwares used for Qtl mapping. First, actually, they used to in 1992. The Lincoln has developed interval mapping, that is actually interval mapping, that is used by one software called as map maker. It is still used actually. It's very easy. And another next came with some composite interval mapping along with interval mapping, like map manager came. Then QGene came. It also do simple interval mapping, and it came with multi-trait analysis in 1997. See how fast they are developing. Then map QTL, and in 1996 like non-parametric mapping and Kursavali rank test. So they are using different I told like nowadays everyone using permutation test to map particular QTL. Again, PLAP QTL came. It can include environmental effect as one of the covariate. It can able to detect environmental interaction. Then MQTL came. Okay. Then different types like QTL cartographer is mostly used actually. It also mostly used nowadays. Still people are using this. It also based on permutation test, you can identify QTL, or a multiple interval mapping you can use for identifying a QTL. Either you can use Bayesian models to identify a QTL. Okay, either you can use single marker analysis to if anything you can do in this software, QTL cartography. Then multi-mapper and ICIM mapping. Okay, these are the several softwares which developed in antis era. Nowadays, I don't know what will come up. Even can do all the things itself. Maybe it will come. Okay. And these are the software regularly we use. Actually, nowadays QTL cartographer and ICIM QTL mapping is regularly used. Then there are like t-test, linear regression, multiple regression, and we should consider a mapping functions, because we know that recombination frequency is not equal to 1 centimorgan is not equal to 1% recombination percent if there is any double crossovers or interference. At that time we had to use correction factor, Kosambi mapping function, Halden mapping functions. It will ask, software will ask which mapping function do you want to use. Normally we use Kosambi mapping functions. Then after learning, and again, what are the factors which affect your QTL map? The genes that controls the trait. If you're having a more or less recombination, the confidence interval you will getting very large. If you're having more recombination possible, the population you're using, if you're using real population, you will get a large QTL, like a large confidence interval. Means your marker is very, for example, you are getting 20 centimorgan interval. Means the markers are located at very far distance from your QTL. There will be chances of further recombination. Your allelic relationship may be going to change. Then the reliability of the selection will become very, very low. To improve the reliability, this year there will be a question on reliability of the selection. For example, he given a recombination. I will tell these things in marker assisted selection. If I tell now, it will get you are getting confused. Always one point you should remember: you should identify a marker which are tightly linked with your QTL. Marker tightly linked with your QTL. If your marker very far distance away from the QTL, there will be chance of recombination further. Then there will be change. For example, you're saying capital M is linked with capital Q allele and small m is linked with small q allele. But if there is a recombination between them, capital M may go with small q, small m may with capital Q. Then this will be good for selection or bad for selection. You understood my point? For example, you are identifying a capital M is linked with a capital Q and small m is linked with small q. Capital Q using red color flower and marker 1, marker 2 will be there. Like say marker small m, marker m. And small q will give blue color flower. But you are saying when there is a capital M1, there will be a red color flower. And small m1, there will be blue color flower. But the marker and Qtl having far distance, there will be chance of recombination. What will happen? Marker 1, small m will come with capital Q, capital M will come with small q. Then if you are selecting small marker allele, what will happen? Blue color is coming. Then you are looking at capital M allele, then you are getting red marker, red color flow. Means your selection will go wrong if marker is far away from the QTL. That's what it's a very much need to identify the QTL, a marker which is very, very tightly linked so that further recombination may not be possible. Like that much near you have to reach. That's why for getting that, we need a more crossing overs like in a population, like RIL. You're getting very, if you use RIL, you'll get a high resolution mapping. High resolution maps. That is one factor. Then heritability of the trait. It's always you think lower the heritability and you'll get, you have to do more effort to identify the QTL because it always affected by the environment. You will not get a stable QTLs. That is the one of the thing. Then because your phenotyping definitely will be affected by the environment. If the phenotypic data will not be good, if you not conducted any multi-environment trials or multi-year experiments, you will not get a good results. That's why if the trait is very low heritability, you should go with multi-year and multi-location trials. Then you will develop an accurate phenotype. Then you will map. If you're not doing, if a trait is low heritability, if you're not doing any multi-year and multi-location experiment, you will end up with nothing. Then size of the mapping population, as I discussed, if you're using small population, you will estimate the QTL with higher effect. But you will overestimate. This is called as overestimation. That's why we should always use a larger population. But how much larger will be again a question. It depends on cost. If you use so much larger population, there will be high costly. And there are several studies, optimum populations, size of population. It again depends which species you are working, the genome size of the crop, either it's cross-pollinated or self-pollinated. So many factors will affect for the size of the mapping population. Then type of mapping population, as I told, more will be the a non-random population generally suited for Qtl mapping. Means you are, see here you are crossing, they are controlled crossing, P1 with P2. They are not random crossing. For example, in a population, random crossing, like it can cross with this, it can cross with this, it can cross with this. If you use that type of population, you will not get, there will be non-randomness. How you will going to get a marker which is linked with the trait? That's why a non-random mapping population generally suit for Qtl mapping. And number of markers you used. Always you remember more will be the marker density, higher will be the higher will be getting Qtl or good maps will going to get. Lower will be the marker density, your lower resolution of the maps. These points you should remember. Means heritability of the trait, size of the mapping population, type of the mapping population, and type of the and number of marker you are going to use. It is very, very important factor which affecting your Qtl mapping. Understood? Any sorry, any points you're not going to understand? Yes, you're not understood.
Sir,
Sir,
what is difference between resolution and power?
Sir, also I'm having same doubt, sir. I will go with them, sir. There
Yeah.
are two terms given, sir. Power of resolution and power of mapping, sir.
Yeah, actually power and means they are normal terms only, but in mapping, I will tell what is the difference actually. For example, you are crossing a P1 and P2, you are generating a mapping population. For example, you are going with the resistance, means you are crossing with high resistance with high susceptibility. You want to identify a QTL for resistance, means the probability of getting QTL in this population is high or low, because you are crossing highly resistant and size susceptible. They are having the resistant alleles and susceptible alleles. They will recombine and they will give the recombinant individual here. The probability of identifying the QTL for resistance is high or low in this particular population.
Hi, sir.
High. Then power is high. Power of identifying the QTL is very high. Then there will be another situation called as association mapping. We are using random populations. We are not creating any mapping population. Here, identifying the resistant QTL from this random population is high or low
Low, sir.
probability.
Low,
Low.
sir.
Then that's why power is low.
Sir,
When come to resolution, here there will be a very limited recombination. Means you will get QTL and marker having QTLs, but it may get 20 centimorgan. Window will be very large. The confidence window will be very large. But power probability of identifying a QTL is very high, but you may get larger intervals. But here resolution is less or high. You are getting a larger window, but
low
you are getting
resolution.
low resolution because you are getting larger interval. You are getting a larger chunk of the genome. There will be a lot of genes may present here. Either the genes associated with a trait or may not associated with your trait. We can't do any fine mapping here. But here that's why resolution will be very low. But in case of association mapping, it takes the account of evolutionary recombination. I will explain because I not dealt with association mapping. Those who are read, you can understand. Those who are not read, don't worry. I will explain. There will be evolutionary recombinates. There will be, because we are using a random population, they are having recombination from several thousand years. That's called as evolutionary recombination. Because as I told, more will be the recombination, more will be the resolution. Means you are getting a single marker is very much near to the QTL. You are getting a very smaller window here. That's why resolution is very high, but power is low in case of association mapping. But here, the power of identifying QTL is very high, but resolution is very low. Understood now what is resolution? Actually,
Yes,
I
sir.
thought to discuss these points when I was discussing association mapping, because without understanding association mapping, because you already listened my classes, that's why you're asking doubt. I know. Those who are not read about association, like understood, don't worry. I will discuss association mapping. What are the advantages and disadvantages there? Okay. Then what is
Sir,
okay? Everything is good. Okay, what's your doubt?
epistatic effects can only be estimated to QTL mapping, sir, not through association mapping, isn't it, sir?
No, you can identify epistatic markers, but you have to do a lot of analysis. You'll get. Don't. There will be different softwares are there. Okay, epistatic effects. Then that I will deal if I deal with association mapping, because now if you're asking that doubt, so many people will confuse. So what they are discussing, they will leave the class. Okay, that's why. Uh, please ask the doubts before the topics which are discussed before, not after. Like which the topics is going to discuss, don't ask the doubts after. classes. Only things which I have discussed. For example, I explained these things, like all the things which I have discussed now. If you have doubts in this, you ask. Okay. In future classes, you don't ask. I will discuss them whenever the time comes. Like after discussing association mapping, we have to discuss. We have to compare them. Candidate gene up you have to compare linkage mapping and association mapping. When you get a comparison, you will get you're going to discuss what are advantages of this one, what is the disadvantages of Qtl mapping, advantages. We will comparing in terms of association mapping. Then we will get an idea how to deal with that. Then there is another term called as Qtl support interval and Qtl hotspot. What is Qtl support interval? For example, you are identify Qtl from this region to this region. Means it's a range from this to this range. It is called as Qtl where particular from which range to which range where Qtl is present. It is called as Qtl support interval. What is Qtl hotspot? Means it's a region of the genome having multiple QTLs. QTL1, QTL2, QTL3 like this. Then, and maybe it was good, for example, for plant height, one QTL is there. For flower color, one QTL is there. For yield, there will be one QTL. But they're all located in the single region. If you transfer this QTL to any other variety which is not there, you'll get bumper improvement. So much improvement. That is called as QTL hotspot. So many QTL hotspots are identified. You just search one landmark paper, Qtl hotspot in chickpea by Rajiv K. Varshini. You read that paper, you're going to get everything about Qtl hotspot. What is the importance of Qtl hotspot in plant breeding? Then, it's a high genetic activity. Like normally, how we can typically measure in a Qtl position, like at what position the particular Qtl is there, means it is just Qtl hotspot is just to identify either there is a presence of multiple Qtl there or not, and it having a several traits, genomic region which governing several traits, they are overlapped. Maybe they were occurring in the families. Maybe they having some additive effect on each. Maybe there will be several things. Okay, but you just remember what is support interval and what is Qtl hotspot. Then what is the application of Qtl mapping? Can used in the marker assisted selection. If you're identifying marker with some QTL, you can use it for indirect selection of this particular QTL. Then you can use in marker assisted backcross selection. You can use it for foreground selection. Then you can use it for marker recurrent selection. For example, you're having these very small QTLs. In one individual, one QTL1 is there. In another individual, QTL2 is there. In another individual, QTL3 is there. But you can bring all the QTL into single population so that we can enrich the population with all desirable QTLs. This is called as a population improvement by QTL pyramiding. Okay, there was one ARS question was asked, how will improve by using Qtl pyramiding? We will discuss while discussing marker recurrent selection. I will use Qtl pyramiding, improving the genetic gain of the crop for 15 marks. This is how they will ask very different questions. Then we can use marker assisted pyramiding, gene pyramiding. We will going to discuss, and it can used in gene deployment and genome editing. And if you are identifying any QTLs, then you go for fine mapping of the QTLs. Then you can identify the genes, candidate genes. Then you can edit them. There are several approaches. Normally, all the things will start with identifying the QTL or identifying the MTS. And even though GWAS is more used, but the relevance in the plant breeding is contributed by the QTL mapping. Nowadays, QTLs are regularly used in the plant breeding rather than MTS. We'll discuss why it is. Okay, and after you identify the QTL, there will be two terms: QTL confirmation and QTL validation. QTL confirmation and QTL validation. QTL confirmation and QTL validation. For example, you have taken parent 1 and parent 2 and you have generated mapping population 1 and you identified the QTL. You generating, for example, this is same rice only, but you have created another population. This is F2. Same with same parents, P1 and P2. You generate mapping population, but it is F6. Same parent, you just advance and you develop F6. And this will become a real. And you just test this QTL in this F6 population. You are getting the QTL at same position with the same effect. Then you say, okay, QTL confirmation. This is called as QTL confirmation. Then there is another term, Qtl validation. For example, here P1 and P2 you are using, you use another parents, P3 and P4 in rice, and you develop the population for particular same trait only. For nematode resistance you take. Here also you are looking at nematode resistance. In this population in India, you use identify this QTL. But in some other country, they have used P3 and P4, but they are nematode resistant for same trait, and they develop population. If they check this QTL in this population, again they get same effect and same position. This is called as validation, your QTL validation. Means your particular QTL you identified, it's very reliable for the particular trait and it is validated also. This is how we have to do QTL confirmation and QTL validation. Actually, my study which I show you, it's a QTL validation study. Means I had to the QTLs which are identified from various studies. Then I checked either these QTLs present in my population or not. Present in my population or not. Because none of the QTLs are not present in my population. We need to go with QTL mapping in to identifying a new QTL map, new QTLs. Because our population not contained any other QTLs for the nematode resistance, I had to search a new QTLs. That's why I had to go for QTL map. But if you are identifying any QTLs which are previously reported in our population, no need to go for QTL mapping again. Because you already QTLs are there in the population which are reported by others. You directly use that marker and you use it for marker set selection. This is why we need to go. Like it's called as QTL previously reported validation of previously reported QTL in the population. Before every QTL mapping approach, first you need to validate any previously reported QTL is present in the population or not. It is a must and should approach before QTL mapping. Those who did, they know already, but those who are not did, please get to know.
Sir,
Yeah.
sir, if we use different parent, then also the population contains the same QTL.
Maybe, may not be. In my population, it's not contained any other QTL. Maybe your parent having a novel genomic region, which earlier it was not there. See, that's why how I published paper, a novel source for root knot nematode resistance, and it's validated by single marker analysis, like trait specific QTL survey. Then I can say that none of the QTL present in my population. It may be a novel source.
Sir, which type of population you used in your work, sir?
I used F2 derived
Okay.
F2 derived F3 pump. This is what application of QTL and QTL confirmation, QTL validation, QTL hotspot, QTL this one. Okay. I hope you got what is QTL mapping, different QTL mapping methods. So tomorrow we will discuss what is QTL fine mapping. Okay. Tomorrow I will discuss QTL fine mapping. What is QTL fine mapping? What are the approaches of QTL fine mapping? What is the use of QTL fine mapping in plant breeding? We are going to discuss tomorrow. Any doubts up to here?
Sir,
Yeah.
sir, if we identified a QTL in some population between two parents, P1 and P2,
Yeah.
then we have to use that same parents in marker registered selection also, or the QTL can be uh, that that be found in that mapping population. We
Yeah.
can use in any other population also. I mean
You
any
can
other
use it to any other
parents also.
population. First, before that, we need to validate. I
Okay,
told,
sir.
na,
Sir,
because
only after validating that becomes a potential QTLR, sir. Otherwise,
yeah,
our
yeah.
work.
Before validation, how you can use that markers? First, you have to check either the marker is polymorphic between the parents or not. For example, if author A has identified on QTL and he developed for QTL is smart, it's markers only, marker 1 and marker 2. You, you check this marker and marker 2 in newer population, parent 1 and parent 2. If it is between the, if it is polymorphic between the parent 1 and parent 2, then only you can use in the population. Okay, you will get polymorphic. Then you have to do validation. After validation only you can able to use that marker in your this one. You can use if you identify QTL in M population, the marker which developed marker which are linked to QTL, you can use it for selection in different population. Only after validation. Without validation, you can't use. Okay.
Sir, another doubt.
Yeah.
If we select two diverse parents,
Actually, now
then
these things I will discuss in marker assisted selection because you
okay, sir.
don't know the schemes like foreground selection, marker assisted background selection. What is selection by using markers? There will be different things. Okay, how to
Sir,
select
this
the
question
marker
is about
allele?
marker.
Yeah, yeah.
Sir, if
Ask us.
if we choose two diverse parents, sir,
Yeah,
then they will be differed for so many characters, not one or two. They will be there is difference in so many characters between those parents.
yeah,
Then we will get so many polymorphism, na, sir, in
yeah.
markers.
That's definitely, definitely.
Then how can we select that particular marker which is associated with the desirable trait that we are studying about?
See, we are going with a single trait at a time, now. We are doing analysis. We have phenotypic data of what phenotype you want. We are not doing phenotyping for all the for mapping population.
Okay, okay, I
We are
understand now.
going with the phenotyping. I told phenotyping. Which phenotyping you will do? The QTL associated with that phenotype you will get. If you use 10 phenotyping, 10 traits, it will give QTLs for 10 traits. If you use 20 traits, it will give QTL for 20 traits.
Yes.
Sir,
Which
is there
phenotypic
any
information you are going to fit, it will going to get.
Sir, is there any possible way that we can exploit already available data like RISE-3K panel and etc. for QTL mapping
Yeah, yeah, yeah.
exclusively?
There are several software like you can raise there are can get genotypic data because see when before publishing they have to upload genotypic data into website public domain. Like you will get the data and everything. Okay, but we need to have population. We need to develop a very costly approach. QTLs and these are QTL mapping. They're very costly approach. We need a lot of skilled workers, we need a lot of markers, we need a lot of facilities, gels, PCR machines. In our lab, nearly we have to, like my ex lab actually, previous lab, we're having nearly 20 PCRs, so many gels, so many masks, so many peoples. In our lab, in our ex lab, they used to like nearly 60 people used to work for only 2 cents. In our guide, there are 30 SRFs, 30 to 40 SRFs. It's a very huge actually. Yeah. Many. I hope you understood what is QTLs. Tomorrow we'll discuss QTL fan mapping. So that tomorrow will be the end of the QTL chapter, and my friend will take association mapping. Okay.
So,
And
Sunday there will be a class, sir.
Sunday, no, I think no class Sunday.
Okay,
I have
okay.
miss out. I have to go outside. Yeah.
Okay.
Sunday there is no class. But tomorrow I will finish QTL fine mapping. And I hope if everyone is available, then only I will ask him to take the class. See, only 8 to 10 members will be there. Like it's not good for me to ask them because they are spending their time and valuable time for teaching. At least there should be a good response. Okay, that's why if members will be like, I will ask another time if any if everyone can able to attend the meeting particular time, then only I can ask. Other I will only continue association mapping. If I get good response, then only it will be, because I already have slides. It's not a big deal to deal with us. Okay. I hope everyone understood what is Qtl mapping. Tomorrow we'll discuss Qtl fine mapping, so that we'll end the class. Okay, we'll end the Qtl mapping classes. Then we'll start association mapping. I think it will take another 4 days to complete association mapping. Later, I will finish all the MS selection methods. Okay. Bye. Good night.
Good night.
Everyone.
Thank you.